Today I'm going to discuss on the topic chemistry of carbohydrates. The important questions of carbohydrate chemistry which are frequently asked in exams are: classify carbohydrates with example, write biological importance of any two, give two examples of each of the following: aldo, pentose, ketose, mucopolysaccharides, polysaccharides, difference between starch and glycogen, glycosaminoglycans, what are mucopolysaccharides, name the function and composition of any four mucopolysaccharides, name mucopolysaccharides, mention their function. These questions are frequently asked in exams, and today I am going to discuss about these questions and important points which come under the topic of chemistry of carbohydrates. Carbohydrates. Carbohydrates are aldehyde or ketone derivatives of polyhydroxy alcohols or as compounds that yield these derivatives on hydrolysis. Examples of carbohydrates are glucose, which give energy, glucuronic acid used for detoxification, cellulose used for dietary fibers and structural component. Classification of carbohydrates. Carbohydrate is classified into monosaccharide, oligosaccharide, and polysaccharide. Monosaccharide are simple sugar which cannot be hydrolyzed to further simple form. Examples of monosaccharide are triose, glyceraldehyde, which is an example of triose. Oligosaccharides are short chain of monosaccharide which is joined by glycosidic bond. On hydrolysis, 2 to 10 monosaccharide units can be produced. Greater than 3 subunits having oligosaccharides are also known as glycoproteins. Glycoproteins are blood group antigens. Polysaccharides are also known as glycans or complex carbohydrates. They are linear or branched. On hydrolysis, 100 to thousands monosaccharides units can be produced. Polysaccharides are sparingly soluble in water. They are not sweetish and they do not exhibit the property of aldehyde and ketone. Polysaccharides are further classified into homopolysaccharides and heteropolysaccharides. Examples of homopolysaccharides are starch, dextrin, glycogen, inulins, and cellulose. Heteropolysaccharides are present in humans, and examples also known as glycosaminoglycans. Examples are heparin, chondroitin sulfate, keratin sulfate. Starch is made up of amylose and amylopectin. Amylose is alpha 1,4 and amylopectin is made up of alpha 1,4 and alpha 1,6 glycosidic linkage, in which alpha 1,6 is branching. Starch is a storage form of glucose in plants. Glycogen is animal starch. It is found in liver and muscles. It is similar to amylopectin in structure. It is highly branched. It has alpha 1 to 6 linkage. Dextrin. On partial hydrolysis of starch by acid or amylase produced dextrin. It occurs in honey. It contains few aldehyde group and it has mild reducing property. I'll discuss the function of glycogen now. It is used as energy storage and material for glucose and liver. Cellulose is constituent of cell wall. It is unbranched polymer of glucose. It contains beta 1,4 glycosidic linkage. Functions are It is used as fiber and unrefined cereals. Inulin is a polymer of D-fructose. It contains beta-1,2 glycosidic linkage. Thank you. Now I come to the most important part of chemistry of carbohydrate, which is heteropolysaccharides. I'm gonna discuss GAG or mucopolysaccharides now. They are unbranched heteropolysaccharides. They are made up of repeated disaccharides. They are constituent of amino sugar and uronic acid. Amino sugar which is there in GAG are D-glucosamine or D-galactosamine, and the uronic acid present in GAG are L-glucuronic acid or L-ioduronic acid. This polymer is attached to a protein or extracellular protein which is known as core protein, then it is known as proteoglycan, except hyaluronic acid. GAG contains sulfate group except hyaluronic acid. Not one thing, keratin sulfate does not contain uronic acid. It contains galactose instead of uronic acid. GAG is found in synovial fluid, vitreous humor, bone, cartilage. Functions of GAG and or mucopolysaccharides are they act as extracellular matrix, they act as cushion against mechanical shock, they are molecular safe, they lubricate the joint, they are elastic to cartilage. Types of GAG are hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin, and heparin sulfate. They are classified on the basis of uronic acid composition, amino sugar composition, linkage, and core protein nature. This is an important table of glycosaminoglycans and their disaccharide units and their function. I'll tell each of them. Hyaluronic acid, N-acetyl glucosamine and glucuronic acid. Function is lubricant and shock absorber. Chondroitin sulfate, disaccharide unit is N-acetyl galactosamine glucuronic acid. Function is provide endoskeleton structure. Keratin sulfate, disaccharide units are N-acetyl glucosamine and galactose. They do not contain uronic acid, instead they contain galactose. Function is transparency of cornea. Dermatan sulfate, disaccharide units are N-acetyl galactosamine and L-ioduronic acid. Function is transparency of cornea. Heparin, disaccharide unit is glucosamine, glucuronic acid, ioduronic acid. Function is anticoagulant. Heparin sulfate, their disaccharide unit is glucosamine, glucuronic acid, ioduronic acid. Some glucosamines are acetylated. Function, they are the constituent of plasma membrane. So in this table we identified that keratin sulfate do not contain uronic acid, instead they have galactose. N-acetyl galactosamine is for chondroitin sulfate and dermatan sulfate. N-acetyl glucosamine is for hyaluronic acid and keratin sulfate. Heparin and heparin sulfate have same disaccharide unit. Only difference is that in heparin sulfate some glucose. So these are some important points which comes under the topic chemistry of carbohydrates. I have told you some important questions, and I have given you the notes of some important answers of that questions. I request you to go through these questions and answers once more and be prepared for your final exams. And please subscribe my channel.